In this video, let's take a look at what transcription is, when it occurs, and what transcription accomplishes. So to understand that, you might be able to tell from the flowchart that I have written there that the basic function of DNA is to give the directions to the cell on how to make any and every protein that that cell will ever need. So in other words, DNA is kind of like the complete encyclopedia set, A through Z, of recipes on how to make any protein. And the first step in making a protein is that the DNA has to be copied into a different form called RNA, and that's what's known as transcription. Calling the cell cycle, we know when a cell first comes into existence, it's in this stage called G1. And G1 is when the cell is living its everyday life, doing its everyday jobs. And so what that means is that during G1, this is when the DNA can express itself and let the recipes be known to the cell of, what proteins that cell is going to make. And so during G1, this is when transcription and translation, which we'll talk about in a different video, that's when this occurs. Because remember, once the cell moves on from G1, once the cell moves into S phase and G2, these are all preparations for cell division. And so that's not a good time for the cell to be really producing a lot of proteins. But during G1, this is when transcription and translation occur. Transcription is one step in protein production, and what we're saying is that the proteins that are produced by various different cell types are what determine what those cells look like and also determines how those cells function. And so the proteins that are produced within a cell really gives the cell its unique look and function. And so you might wonder, if that's the case, and we have so many different cell types like we were looking at, is the DNA in every single nucleus the same? And the answer to that is yes. The simple answer to that is that the DNA is identical because we're a product of fertilization, right? And so every person is formed when a sperm cell fuses to the egg cell, the oocyte. And when that happens, 23 unreplicated chromosomes from the sperm combine with 23 unreplicated chromosomes from the ovum to give us 46 chromosomes, a new organism, a new human being. And so that's the original cell, the fertilized egg, that all of our cells come from. And so all of our cells have the exact same DNA. So the reason that, one cell will look different from another cell, look different from another cell, and function differently is not that they have different DNA. It's that during G1, what's going to happen is that different segments of the DNA molecule are going to bubble open, exposing a segment which is known as a gene. So a gene is just one segment of the DNA that carries the recipe or the instructions on how to make a single protein. Once the DNA bubbles open and exposes a gene, we're going to copy that gene into a strand of messenger RNA, and that's what transcription is all about. And so the reason that that's necessary, let's take a look at this picture, is you may remember that DNA is located inside of the nucleus. DNA doesn't leave the nucleus. DNA is like the important document under glass. The cell's not going to be able to directly interact with it. So that's where the instructions are on how to make a protein. You might also know that the actual organelle that makes the protein, the ribosome, is not in the nucleus. And so here's problem number one that's solved with transcription. The recipe for how to make the protein is in a physically separate location than where I have to make the protein. That's one reason for transcription. The other reason for transcription is that the ribosome is made out of RNA, which is related to DNA, right? It's a nucleic acid, but it's not the same as DNA. And so when we copy this instruction, this segment of DNA, copy a gene, we don't copy it as a strand of DNA, we copy it as a strand of RNA. And that strand is known as messenger RNA, and what will happen is that copy will leave out of a nuclear pore, go out into the cytoplasm, and attach to a ribosome. So let's take a closer look at that process. Here's a picture from our book looking at transcription, looking at some of the details of transcription. And so all of this, keep in mind, is occurring inside of the nucleus, right? Because inside of the nucleus, this is where the DNA is located. And so they're showing us the double-stranded DNA molecule in purple, and they're showing me that a region is bubbling open that's going to expose a gene. And inside of the nucleus, there are these enzymes called RNA polymerase. And when the DNA bubbles open and exposes a gene, this enzyme will automatically attach to the DNA and start, you can see in blue, constructing a strand of messenger RNA that's complementary to one of the strands of DNA. And when it does this, it's going to use the base pairing rules that we're familiar with. So here I can see they're zeroing in on if this was the DNA template. Okay, in other words, that's the DNA in the nucleus, and then the RNA polymerase comes in and it starts building messenger RNA. That if on the DNA molecule there's a G, then my messenger RNA will use a C. Okay, G binds with C. A on the DNA would typically bind with T if this was another strand of DNA. RNA uses uracil instead. G binds with C, C with G. And once we get up to the top here, we can see A would bind with T, but it's binding with U. And then be careful of this one in DNA where there's a T. Remember, T binds with A. And RNA still uses A. A common mistake would be to put a U there, but U is being used in substitution for T when you make an RNA strand. And so the messenger RNA that's created is just complementary to the original DNA strand. Taking a look at this picture from the book, we can see that the messenger RNA strand is growing in length, that what's happening is this enzyme is moving down the DNA and it's copying a segment of the DNA known as a gene until it finishes and you have your completed messenger RNA strand. In this picture in our book, they're showing a little bit more detail. They're using color coding to show us the base pairing rules, but the premise is the same. What's happening here is this is all, keep in mind, in the nucleus, because here's the DNA molecule contained in the nucleus. Keep in mind that this is not an organelle, this is an enzyme that they're drawing. This is an enzyme that produces messenger RNA, and the way that it does that is it just takes RNA nucleotides, just like a puzzle, and it plugs them in so that if this were an A, then it would use a U nucleotide to go there. And if this was a C, we'd use a G nucleotide and so on. So we're just using the base pairing rules to build a strand of messenger RNA. That is transcription.